Greetings from the 85th Cold Spring Harbor Symposium on Quantitative Biology. As you can tell, the meeting this year is being held virtually, and the topic is biological timekeeping. I'm Brian Ray. I'm a senior editor at Science Magazine, published by the American Association for the Advancement of Science. And I've had the privilege of attending this meeting and learning about how these processes of biological timing have strong influences on organismal physiology and health. And the organizers have generously given me the opportunity to sit down with several of the speakers and have them explain to us some of the most interesting findings that were presented at the meeting. And so to help me do that today is Charlotte Helfrich-Forster from the University of Würzburg in Germany. And I think many people are familiar with the circadian clock, this daily clock that keeps us in cycle with the light-dark cycles as the Earth rotates. But your work, Charlotte, deals with a timing mechanism called photoperiodism. And this is the mechanism that lets animals sense the change of seasons. So if you think about this, this gives animals like the insects that you study a way to follow the seasons and to know when breeding season is, so to help in reproduction. So maybe one of the most important timing mechanisms of all that allows them to do that. And so I think this is a little bit complicated, how animals are able to do this, how they're able to keep track of the day length that's going on, the changes in day length that go on as a year progresses, and respond appropriately to seasonal changes. And so I'm going to ask you, can you explain to us how that mechanism works? And this is a little bit tricky, so the explanation I think will be more than a couple sentences, but if people stick with you, it's a very cool mechanism that you propose for how animals can sense that and do their reproduction at the right time.
Okay, thanks, Brian. I will do my very best, but indeed it's complicated. And probably I have to start with plants and not with animals, because the first mechanism was proposed by Bühning in the 30s of the last century, and he worked in beans, in the common bean, and he noticed that these beans are moving their leaves up and down and very irregularly, and they always reach this kind of day position 4 to 6 hours after dawn, and then the leaves are lowered again, and the night position is then reached 8 to 10 hours after dawn. And this is always the same. It doesn't matter whether the day is short or long. And of course, this means that the down movement of the leaves is not always in light. So under a short day, this down movement is in darkness, whereas under a long day, it is in light. And then, of course, this can be used by the plant to know whether it's early spring or late spring or already summer. And the plants do so. So they need the leaves to be in darkness in early spring, just after they germinate, to trigger the flowering that occurs much later in early summer. And if they don't have it, they produce only very little flowers. So, and he proposed that this may be a common mechanism for all organisms to measure day lengths. And later, it was somehow refined by Pittendrigg, who is, of course, also one of the big fathers of chronobiology. And he termed the entire time measurement the external coincidence model, because the organisms just compare their internal time with the external time. Okay, so his model is a little bit more refined, and it's more working for insects, also flies, but in principle, it's very similar. What is most important, that you have an oscillation that starts in the morning and then have always the same phase. And if a certain phase of the oscillation is then in darkness or light, then the animal can know what time of the year it is. And actually, this oscillation does not have to be very strong. So it's just important that it is continuing under these light-dark cycles. So it doesn't matter whether it continues under darkness or under constant light. That's interesting. And then there is another possibility, and that is called the internal coincidence model. And here, that is actually really from Friedrich. He proposed that there are two oscillations in the same organism. So let's now say in animals, for example. And one of these oscillations always couples to dawn. For example, you can imagine that activity is high in the morning, then comes a siesta, and then comes a second activity bout, and that couples to dusk. And indeed, you see in many animals that they have this kind of two activity bouts, and even the humans, of course, we have this kind of midday, we like to have a siesta, especially in warm days, and especially in the countries, in the southern countries, we really do this if they can. So, and now you can easily imagine that, of course, under a long summer day, the morning activity and the evening activity are far apart. And in between, there's a long siesta, while in a short day, they come close together, and perhaps you even don't need any siesta, and the two activity peaks are overlapping. And now you can imagine that the organism can just measure the time that is passing between morning and evening activity. And that, therefore, it measures a kind of internal coincidence of time. That's the reason why it's called out like that. Okay, and now let's come to flies. Because flies are perfect model organism that may explain this internal coincidence model. And this is because several groups have found that there are indeed morning neurons, so neurons in the brain that work as morning oscillators, and then other neurons in the brain that work as evening oscillators. So they are anatomically defined, and the flies have nicely this morning activity peak and evening activity peak. And what we also find is that under shorter days, the two are close together. Under longer days, they are coming apart. So may work nicely. So actually, it would be wonderful to starting now with hibernation, or we call it in insect diapause, or perhaps better reproductive arrest in flies when the days become short in the autumn, and to see that the flies then go into the diapause and how it's regulated. So for example, in Drosophila melanogaster, we will be able just to eliminate one of the oscillators, and actually everything should not work if you do so. But there is not a single paper about this. And the reason is simple, because the flies, they come from the tropics. We show such a shallow photoperiodic response that it's hard to measure, especially after manipulation of the flies. And that's a problem. And so we had the idea to look at other Drosophila species that live in the very north, and to see whether they also have this morning and evening activity, and then perhaps to see whether that also determines their time measurement. And we did so. We started with 4 species, all stemming from Finland, and we realized that we don't have 2 oscillations. So they just start activity very late in the morning, and then it increases and it extends until dusk. So it's really long. It can even follow a 20-hour long day. And this is, for example, for the tropical species not possible, because they stop activity long before dusk sets in. It's even most activity then in the midday, perhaps not perfectly adapted. Okay, so we were disappointed that the northern flies didn't have these 2 activity peaks. But on the other hand, it was nice, because when we looked at their clock network, we discovered that they have no functional morning oscillators. So perhaps it's not a wonder that we don't show a pronounced morning activity. And the other thing is that these flies became arrhythmic very quickly after we put them in constant darkness, but also in constant light. So it's a damped oscillation, it's a weak oscillator. And we know that these morning oscillators are also very important for robust activity under constant darkness. So that fits perfectly. So they don't have it. And they have something else in their brain. So they have a certain neuropeptide that is able to delay the evening activity, expressed in places where in Drosophila melanogaster you don't see it. So in these northern fly, it's close to the evening neurons. And that can explain why they are active for so long. So overall, it fits nice. But we didn't know whether this is the general principle, because we studied only these 4 species, and later it turned out they all came from the same Drosophila group, Drosophila virilis. So then we started something new, and that's not published, but we really looked for many other species, so 33, not more, but it's a lot, because we have to study their locomotor activity and also brain anatomy. And they came from all regions over the world, from northern ones, southern ones, and even close to the equator, and also some southern ones. And we realized that only flies of the virilis group had this pattern that they lack the morning oscillators. The others seem to have both oscillators. And most of them have also these extra neuropeptide fibers close to the evening cells. And what was very nice, we found a wonderful correlation in the distance between morning activity and evening activity and latitude. So the farther they are far away from the equator, the more they could extend morning and evening activity to dawn and dusk. So that would actually, it speaks for this internal coincidence timing. But it's just the beginning. We know that we all do diapause, but we have not studied, of course, the role of these oscillators in initiating diapause. So that would be the next step, and it's more complicated because these species, they cannot easily manipulate as we can Drosophila melanogaster. So that was a long explanation. Please tell me whether something is still open.
No, that's amazing, and I just think it's such a beautiful story, because you've taken the work from your lab and many others to understand the anatomy of the fly brain, to understand where these particular groups of neurons are that are controlling these individual clocks that are cycling at the beginning and the end of the day, and you have the ability to manipulate those to control them and see what happens. But then you also had this clever idea to compare animals that were in one part of the world and another, and you can actually see differences in the way the brain is connected in those flies, so that they've adapted to be able to live in the environment that they find themselves. And so they adjust this timing mechanism where they're measuring day length to be appropriate to the particular part of the world where they live. And I just think the way the science extends all the way from these individual neurons and your ability to map them to the ecology, to the life cycle of these animals is just a remarkable story.
And what I think is very nice, that obviously the animals have taken several ways to adapt. So it's not always the same. So probably if we look in more detail, we will even find more ways how they can adapt. And of course, I also have to say the clock is important, but of course it's not the only thing. They need many more adaptations to really adapt to such a life in the north. For example, with Forster, at least cold resistance. And that's something, of course, it is tempting to speculate that also the clock triggers that, but we have to show it.
And you get challenges as you move, as you move out of the Drosophila melanogaster that is so amenable to whatever you want to do, and you may not have those same experimental possibilities in these other species. Yeah. All right. Well, thank you so much, Charlotte, for taking the time to explain that. As I said, I think it's a wonderful story, and we appreciate your cooperation and taking the time to be with us today.
So it was a pleasure for me. Thank you very much.